Welcome all 103 people who are joined to this meeting. So we were taking bets before, and yes, the person who said it would be more than 101. So congratulations. So welcome to the CHESS Commons meeting. This is a special edition today. The highlight of the meeting today is Rick Cerione talking about COVID-19 research. So I'll hand it over to Joel.
Good morning, everybody. It's a pleasure to see you here today. Or I guess you can see me, I can't see you. So, as you're all very much aware, almost all research activities on the Cornell campus have been suspended since mid-March. And the exceptions have been for essential maintenance and research directly related to COVID-19. The university set up a process to apply for these exceptions, and a committee, of course, was established to review them. And of course, they were also imagining applications from individual faculty members requesting lab access for just one or two researchers. And as everybody on this call knows, that restarting a synchrotron facility is a much larger endeavor than that. And many of you have already been involved in developing the detailed plans that are necessary to do it. In the end, we submitted a proposal, and the committee that reviewed it first kicked it up to the vice provost for research, who decided that he needed to kick it up to the provost, who then, not surprisingly, consulted with others, presumably the president in Day Hall. But after all of this consultation on Wednesday morning, we finally received notification that our proposal to restart the facility to conduct, in quotes, research that is essential to the COVID-19 mission was approved for an immediate start. So just briefly in summary, we've been approved to operate the facility to perform a single X-ray experiment on some specific drug candidates. And of course, we're going to need to strictly follow social distancing guidelines, and personnel safety is a key issue here. So with that, I will yield the stage.
Okay, so I guess everyone knows that Joel strong-armed me into giving an overview of the work that really has been going on in my laboratory and how we think we would greatly benefit from being able to do this together and ultimately work together with CHESS and McChess. And so what I'll do is give you just a brief overview of what we have and what we're planning, and then just give you a little bit of background and rationale for the drug discovery that we're doing. And then I'll briefly, because most of the drug discovery that we've been doing is targeting cancer, and this has been the case for some time now. And then the similarities actually between cancer and viral infection, and then what the current and future goals are, and especially again, taking advantage of CHESS and McChess. So in terms of the overview, our laboratory now for some time has been developing and trying to develop new classes of anti-cancer drugs. And now it's gotten to the point where Cornell has enlisted a venture investment group to help us with funds for this drug discovery. And we're targeting metabolic enzymes that are essential for cancer cells to be able to survive and act like cancer cells, and whereas we don't want to have any effects on normal cells. And we need structural information to better understand really how these drugs function. How is it that a given drug is very potent or efficacious, and another one that looks a lot like it is much less so? We really would like to be able to get some mechanistic and molecular details as to how these drugs are working, because we want to keep developing them. And we know there's going to be another wave. We know there's going to be another pandemic. And we know NIH is going to be skewing all of their money towards these sorts of things. Now, the problem has been for us is that standard X-ray crystal structures haven't, in many cases, allowed us to do just that. They haven't, the structural images we have of our candidates bound to our targets, we might have two drugs, and you'll see some examples of this, that differ quite a bit in their potency. But when we look at the standard cryo-cooled X-ray structures, they look virtually identical. And so this is where we're really hoping that the new development, serial room temperature crystallography, will allow us to tweeze out the differences between a very potent drug and a much less potent drug. We have ideas based on chemistry and biochemistry what makes something more potent, but we'd really like to confirm this and learn more about this. And it comes down to some real subtleties. And that's where we're hoping that this room temperature crystallography will allow us to see that. And then importantly, it turns out that the same enzymes that we're targeting with our anti-cancer development, these very same enzymes are being used by viruses. They're being used by SARS-CoV-2 or COVID-19, and thus the relevance. So I'll just give you a little bit of a background and rationale for the drug development we've been doing, and then I hope that'll at least give some level of relevance as to why we want to pursue this structurally with Chess and McChess. So it turns out that for some time, our laboratory, what we've really been interested in are basic questions. I mean, we want to understand how cells grow normally, the signaling pathways that cause them to grow normally, and then what goes wrong in those pathways when cells stop acting normal and they become cancerous, or in the case of the nervous system, when you get neurodegeneration. And so we were doing a lot of quite basic research. And then along the way, we started thinking about ideas about how could we design or identify small molecules that would selectively block the things that are going wrong in a cancer cell without affecting a normal cell. And through those efforts, and it took some time with a lot of students and postdocs' work, we came upon a group of compounds that look very, very promising in terms of their ability to make aggressive cancer cells start behaving like normal cells without affecting normal cells. So that's always something that, at least at the first level, is attractive. The surprise came when we really figured out how are these small molecules that we think are drug candidates, how are they really working? Because we assumed initially they would be working by interrupting signals that cells use to grow or grow abnormally. But it would turn out that these small molecule drug candidates were really impacting the basic metabolism of cancer cells. And so without going into the gory details, all this really represents is that if you look on the left-hand side where it says normal cell, in normal cells, you have a pathway that's in the soluble part of the cell, the cytosol. It's called the glycolytic pathway. So it starts with when sugar enters the cell, glucose, and then there's a series, step-by-step, of enzymatic reactions, and the end point of that pathway produces a metabolite. It's called pyruvate. That metabolite then enters the energy organelle of cells. This is called the mitochondria. And when it enters the mitochondria, the pyruvate's converted to another metabolite. It's called citrate. And that metabolite starts a cycle of enzymatic reactions in the mitochondria. This is sometimes called the TCA cycle. And each enzymatic reaction in the cycle produces some number of ATP molecules, ATP being the basic energy molecule of life. And so it's this coupling between this glycolytic pathway and this cycle that pretty much satisfies all bioenergetic requirements of normal cells. But what happens in virtually every type of cancer cell is that this glycolytic pathway is uncoupled from the TCA cycle, such that the end product in glycolysis, pyruvate, in cancer cells, instead of being converted into mitochondria to citrate to start this cycle, the pyruvate is almost entirely converted into something else called lactic acid. And that stays in the cytosol, and that happens because cancer cells have a special enzyme to do that. And actually, there are reasons and benefits to the cancer cells to uncouple this glycolytic pathway from this cycle. But nonetheless, cancer cells need to find alternative ways to feed and turn this cycle, not even so much for making the ATP, but to make building blocks for biosynthetic processes that cancer cells need to be malignant. And the way they find the alternative, or the alternative they develop, is by greatly increasing their ability to metabolize an amino acid called glutamine. And so cancer cells, in fact, are said to be glutamine addicted. And that happens as an outcome of a greatly accelerated first step in glutamine metabolism, this conversion of glutamine to another amino acid, glutamate, by enzymes we'll talk about in a moment. And then that product of that reaction is converted by yet another enzyme into another metabolite that feeds this TCA cycle so it turns. So this is how cancer cells compensate for the uncoupling of this glycolytic pathway from the TCA cycle by increasing their ability to metabolize glutamine. And so what we've been targeting are the enzymes that catalyze that first step in glutamine metabolism. They're collectively called glutaminase. Now, it turns out humans, mammals, have two forms, two genes encoding members of this enzyme family. And one form is called GLS, or sometimes GAC. The other form is called GLS2. And these two enzymes show up very, very highly expressed, very highly activated in a number of different cancers. So GLS, as an example, highly expressed, highly activated in triple-negative breast cancer. GLS2, on the other hand, is the predominant form of the enzyme that's highly activated and expressed in progesterone receptor and estrogen receptor positive breast cancer, which makes up about 70% of all breast cancers. In ovarian cancer, it's GLS2 that's highly activated. In uterine cancer, it's GLS2. In brain cancer, it's GLS. In colon cancer, it's GLS. Some cancers use both of them. They catalyze that same first step in this metabolic pathway. And the same enzymes are being used, highly expressed, highly activated when the virus infects cells. Okay, so how have we been trying to attack this enzyme that's so important in cancer? So we've learned a fair amount about how this enzyme becomes active. And it turns out in normal cells, there's very little of this enzyme. They're not glutamine addicted. So it's very poorly expressed and it's very weakly active. And under those conditions, this enzyme really isn't doing much of anything. It's a dimer. But in cancer cells, when it gets highly expressed in the cancer cells, and that happens incidentally because cancer cells have cancer-causing genes that cause the cells to become cancer. They're called oncogenes. And they send signals to call up the expression of this enzyme and to make it very highly active. And when that happens, it transitions from this inactive dimer, as you would find it in a normal cell, to an active tetramer. So in this right panel, this is a space-filling view of the tetrameric form of this enzyme. So in dark blue and light blue in the top half, that's one dimer, and then light blue, dark blue in the bottom half is another dimer. So this comes from X-ray structural work. It's been done. Ching Chu has done a lot of this. Postdocs and students in my lab have done a lot of this. So the first, let me go back. The first class of compounds that we identified, the lead, we just code named it and called it 968. And this is a compound that will sit in a cove that forms between these two subunits that are making a dimer of this enzyme. And when 968, or the analogs we now have, sit there, it prevents that dimer from receiving signals that are only happening in cancer cells from these oncogenes that would convert it to a tetramer. So this small molecule and the analogs that we have now trap the enzyme as an inactive dimer. Now, the other class, there's a second class of inhibitors. The lead compound is called BPTES. This was actually first identified by a biochemist at Colorado State who was looking at not disease or cancer, just other processes. And then it turns out a biotech company on the West Coast called Calithera developed what they called a more potent analog of this. They call it CB839, and this is now in clinical trials for triple-negative breast cancer and for multiple myeloma. And then our lab has also developed more potent analogs. So the Calithera group has changed this lead compound at the ends, and then our group has changed it in the middle. And when ours is also very potent, but we think it's more long-lasting in people than this particular compound. Now, these work in a different manner from the 968 class. They actually bind in the interface in the middle of this tetramer where the two dimers are coming together. And you get two such compounds bound per tetramer. And when they bind, they prevent the movement of a loop that happens in each one of these subunits that sends a signal to the active site to open it up so you'd have catalysis. So these molecules prevent the loop from moving. They don't let the active site open, so they trap it as an inactive tetramer. This class traps it as an inactive dimer. Now, it turns out that these two classes of inhibitors positively cooperate. So we found that this class, when it binds, enhances the binding of this group, and this class, when they bind, reciprocally enhance the binding of these inhibitors. And this is rather unusual in drug development in cancer, because what most drug companies are doing is that they pick out a particular target. It can be an enzyme, it can be a protein kinase, and different companies design different inhibitors, but they all bind at the same site and act at the same way. So you don't get much, in terms of a combination, you don't get much of any benefit more than just increasing dose. But these can work cooperatively. So these raise some real possibilities in terms of a cocktail where you would use both classes of inhibitors. And let me just give you one example. So here, we were looking at a really nasty, aggressive brain cancer cell, growing it in the laboratory. So this first histogram that's dark, just untreated, those cells are growing like mad. The next histogram, we were treating with an analog of that 968 class, one of our better analogs. But at this level, it really isn't doing much of anything in terms of touching that nasty glioblastoma cell. This is CB839 on its own, also on that level, not doing much. But if we combine the two, because of the positive cooperativity, we start to see a significant inhibition. And we can push this down really pretty far. So this is going to be something we're going to be very interested in further developing. Now, we've been developing a number of analogs of this 968 class. And what this amounted to was first to develop a fluorescent readout with the purified recombinant enzyme. So we put a reporter group close to where these drugs bind so we can watch when they bind because they quench the fluorescence. So we can get dose-dependent binding. And then we developed a spectroscopic assay for enzyme activity. So now in the same cuvette, simultaneously, we can look at the binding of different analogs to see how well they do and their effects on enzyme activity. And that's what's being shown for some of these in the lower left. And you can see there's really a perfect correlation. The ability of a different analog of this inhibitor to bind directly to the purified recombinant enzyme matches its ability to inhibit the enzyme activity. And if we overlaid on top of that, the ability of these different analogs to inhibit cancer cells, that would fit perfectly. But in the lower right, you can just see that there's a range. There's really a range of these compounds, although they all look very similar in terms of their potency. So again, that's what we want to better understand to ultimately do better drug development. Now, we've been using these compounds to test them in mice, giving mice tumors and then introducing the compounds. And these 968 analogs that are the more potent work well in inhibiting the tumors in the animals. But one of the real problems in cancer is resistance. So cancer cells are devious. So you block one step in the cancer cell, one pathway, and they find ways around that because they have numbers of oncogenes firing at the same time. Cancer is a multi-oncogene problem, and they keep finding workarounds. So what we've been doing more recently is what we're trying to do is use drug combinations where we use one drug that would make the tumor cells at the primary tumor site really hypersensitive and hyper needing these metabolic changes so that if we come in with the metabolic inhibitor, we really whack them and don't give them the chance to start up and ultimately give us resistance. So let me just give you an example of that. So what we're doing in this experiment, this is a mouse experiment. So we've taken triple negative, it's what's called patient-derived tumor grafts. So you get triple negative tumors from patients and you can graft them into mice. And then you wait a few weeks and you start to detect the tumors. And then you start to treat with compounds of interest. So what we're looking at here is one of those sets of experiments. The growth of the tumors in the animals is a function of time. So in black, this black line, that's just the animals starting to show these tumors taking off. They're untreated, the ones in black. Now what we've done in this combination is we've combined the metabolic inhibitor I was talking about, 968, with an FDA-approved anti-angiogenesis drug called Avastin. Now, this was a drug that was developed some years ago by Genentech, and it came with great promise because the strategy was to block the ability of tumor cells to receive a blood supply. So these cancer cells at a primary tumor site send signals to get the building of blood vessels to provide blood to the tumor and nutrients, and this is called angiogenesis. So this is an anti-angiogenesis drug. As I said, it was hailed with great promise. It's still in use today in the clinics, but it has its own limitations as a single treatment agent. But in these experiments, so if we add just that drug, Avastin, we're getting some slowing of the tumor growth relative to animals that aren't treated, but it's far from a complete response. Now here in green, what we're doing is we're introducing a really low level of this metabolic inhibitor, 968. And in fact, at this level, it almost helps the tumors in the beginning, but then you really get no benefit. It really doesn't do much of anything. Now, if we increase the dose from this very low level of 968 and keep upping the dose in the mice, we'll start inhibiting the tumors, but not at this level. However, if we combine this level of the metabolic inhibitor and this level of Avastin, we get this. And so we think this sort of combination, the Avastin starving initially the tumor cells, caused them to be hypersensitive to this drug or this treatment. And that's why we think we're seeing this pronounced synergy. And we've seen this in a number of examples now. So this is just another set of experiments. Again, it was triple-negative breast cancer from a patient into the mouse. And you can see the combination of this metabolic inhibitor and Avastin in this lower right really having a profound synergistic effect. And it's this that then got these investors really interested in trying to go further in drug development as anti-cancer drugs. But we've also been interested in that second class of allosteric inhibitors, this BPTS CB839 class. And so to develop those better, we've done the same thing. We developed a readout so we can monitor the direct binding of the drugs to the purified recombinant enzyme. We've developed a readout so we can read out the effects on activity. So here, this is the binding assays, and this is a semi-log plot. So here's the lead compound, BPTES. This is CB839 that's now in trials. And this is a compound we developed that we think, at least in the animals, is going to do a little better. And again, you can see from a chemistry point of view, CB839 is a little different on the ends relative to the lead, and we're a little different in the middle. In terms of direct binding to the pure enzyme, the black circles, this is BPTS binding. The red is CB839. So you have a dose that's shifted. And again, this is semi-log. So it's a significant increase in potency, as was claimed by Calithera. And then this is the one that we've developed, and it's also very highly potent. And we've done the same thing looking at enzyme activity. So here we're just comparing the drug we've developed with the lead, and you can see in inhibiting the enzyme, the activity of the pure enzyme, a dramatic difference. And again, this really gets to the crux of our challenge, because we have crystal structures for these two drugs bound to the pure enzyme, and they look identical. So there's a significant difference in potency, and we want to understand what is causing that difference so we can keep making better drugs. And again, that's where we're hoping we might do better with room temperature crystallography. Okay, so where does the virus come into all of this? How does SARS-CoV-2 infect cells? Now, many of you may be well aware of this. I just pulled this picture out of Time magazine. So this is supposed to be for the lay public. And basically, the way this virus works, and again, forgive me if you all are well familiar already, but it turns out this is what's called an RNA virus. And it has an envelope surrounding it. Oops. And on the envelope are proteins that look like spikes. This is where corona comes from. And so these spike proteins, or S proteins, are what allow the virus to bind to a receptor that's on the cells they're infecting. That receptor is called ACE2. And what has to happen for that is that there has to be a limited proteolysis, a little clipping of this S protein, so you can expose the residues that are going to do the binding to the receptor. And then the virus bound to that receptor, most people argue, is taken up into the cell by a process called endocytosis, which cell biologists are well aware of. And it's when the virus gets into the cell that it starts to take over the cellular machinery to make more virus. And in taking over the cellular machinery, just like oncogenes, when they're making cells cancerous, send signals to change the metabolism, the virus has proteins that can cause the host cell to change the metabolism in exactly the same way. And so then what happens is that if you make enough virus and more virus, these viruses start to bud off and leave to look for new host cells to infect. And usually this cell will end up lysing and dying. So, what are the similarities between cancer cells and viral infected cells? So they both have to reprogram the metabolism. The cancer cell is doing it. Oncogenes are causing a cell to become cancerous and change its metabolism. Viruses that infect cells are causing the host cell to change their metabolism. An important reason for this is among the things that these metabolic changes in cancer cells do is make small vesicles, roughly the size of a virus. These are called extracellular vesicles. They're sometimes called exosomes. So the cancer cells have a new metabolism. They're making these small vesicles that they then shoot out. And those vesicles do some really important things in malignancy. The first thing those little vesicles do that the cancer cells shoot out is they impair an immune response. So it turns out that those little vesicles on their surface have something that can bind to T cells, which is the white cells that are going to hopefully would attack the cancer cell and destroy them. But when these little vesicles bind to the T cell, they destroy the T cell. And so this is the whole sort of strategy of immune therapy that's getting a lot of recognition in the literature now, and of course in the media, is that they are developing, and James Allison, who won the Nobel Prize 2 years ago in medicine, developed antibodies to block the ability of these little vesicles to bind and impair T cells. This is what immune therapy is. The other thing that those little vesicles do is they're coated, and depending on what cancer cell makes the little vesicles, they're coated on their surface with different proteins that target them to different sites. So breast cancer cells, for example, will make these little vesicles that'll go to the lungs, sometimes to the brain. Prostate cancer cells will make them, they go to the bone. And when they get to these second sites, these little vesicles change that environment of the secondary site so that the few circulating tumor cells, the tumor cells that break free from the primary tumor site, get into the circulation. They get to these secondary sites that have been made welcoming by these little vesicles, and that's how metastasis starts. And it turns out that the virus, when it infects the host cell, changes the metabolism, and that does the very same thing, makes these little vesicles that are almost the size of the virus, sends them out of the host cell. It's being argued now that's impairing, making it more difficult to get an immune response, and they go to other places to make other cells receptive for more viral infection. Now, it turns out the ability of these little vesicles to be made in cancer cells and in virus-infected cells, there's a trafficking process. They get produced, they have to get trafficked. That can be blocked by a drug that's been in the news quite a bit called hydroxychloroquine. I was going to say something about our president, but if this is being taped, I'll behave myself. But this has been purported as being a miracle drug by some. It's an anti-malarial drug. It has its benefits, but I think if you've seen the news, you've also heard that its dosage, high dosages, can be very dangerous for patients, particularly with cardiac disorders. But before we even got into the virus work, because we knew that this hydroxychloroquine could interfere with the vesicles that were being made in the cancer cells in response to the metabolic program change, that we thought, what about combining metabolic inhibitors with hydroxychloroquine? So we did this in cancer cells, and this is an example. So again, we have this is another really nasty brain cancer cell. So they're growing, and they show really good survival under all kinds of stress. That's part of the problem with those cancers. And so this blue histogram, untreated, the orange treated with some level of 968s, not touching this aggressive brain cancer. Treated alone with chloroquine, not touching, but look at the two together. We get this really dramatic synergy. And there's interesting ideas among the lab as to the underlying basis for how you get this pronounced synergy. But it was this that really got us fast-tracked at Cornell to look at these compounds and these combinations against COVID, because the argument is if you can get the doses dropped because of the synergy with chloroquine, then you might have a real benefit. So we're wrapping up here. So current and future goals. So the lab, my lab in molecular medicine, what they're doing is that they're making what are called pseudoviruses to mimic SARS-CoV-2, but these are viruses that are missing one piece that doesn't allow them to be transmitted. So basically they're the virus, and then we can coat that pseudovirus with these spike proteins. And then we can add those viruses to a host cell. And it'll get into the host cell, it'll change the metabolism of the host cell, but it's not able to make enough new viruses, and it's missing the piece that will allow them to be spit out and really become infectious. So you can do these experiments in BSL-2. And so what we're doing is we have the host cells that are the parent host cells and the host cells with the pseudovirus, and we're looking at doses of our different metabolic inhibitors singly, in combination to two classes of metabolic inhibitors, and then in combination with hydroxychloroquine. And then based on those results, return those over to a virologist we're working with who's here in the vet school, Gary Whitaker, who's growing human SARS-CoV-2 in his BSL-3 facility. In fact, he's one of just 20 sites in the US doing this. And so based on what we find, he's going to take the best dosages and combinations that we think we have, and he's going to then test it on the human virus. And then based on his results, they come back to us, because this is going to be an iterative process. We're going to have to do some refining, and then back to him. Okay, so in chemistry, and this is where chess comes in, there are people in the lab that are now preparing complexes between the pure target enzymes and these different metabolic inhibitors, singly or to two classes in combination. And it's here that we hope that working together with Aaron and Arena and Chess and McChess, through room temperature crystallography, we really can learn more about how these drugs are working, because this will help us initially be able to design better ways to use them, better doses, better combinations. But the other thing it's going to be really critical for is down the road, because as I said, we know there's going to be another wave coming, and we want to do better drug design. And we're hoping that serial room temperature will let us do that. And so in many ways, independent if our compounds ever work to cure anything, it's a test case, it's a proof of principle. And because we have so much biochemistry and biology that backs up the structural work, this is a great system, I think, to develop the serial room temperature crystallography. And our hope is if we really do, we're going to attract a lot of other people who want to come here, who will have their targets and will have their drug candidates to do better rational drug design. And I guess the final important thing is that these experiments that will be happening at Chess and McChess do not involve infectious agents. We're not bringing in virus. We're bringing in pure proteins that we were bringing in for years, and drug candidates to learn more about how to do a rational drug design. So I think it is really an exciting time. I think there are great opportunities. I think it's a direction that Chess and McChess can take advantage of. I think we're going to try to take advantage of this every way we can with funding. So that's my overview, and I'd be happy to answer any questions, which I'm sure I won't know the answer to. But
I have a couple for you. Two from Ken Finkelstein. He's asking, does this strategy to treating cancer require patients to continue taking the drugs forevermore?
Yes, and we don't, the answer is we don't know. I mean, what we hope is that when using these drugs, that we're actually returning the cells to their normal metabolic program. And then the question becomes, if there's a continuing insult from oncogenes, then you're going to keep trying to drive that normal program back to a cancer program. So it's probably going to have to be a combination with other compounds, ultimately, that will prevent the oncogenes from working so that you can get these cells back to normal, and then you can do something to prevent those oncogenes from trying to convert it back to a cancer cell. So I think, the investors love the idea that people have to take these for life. I've learned a lot. I've learned how naive I am. I've learned a lot about how drug investors think and what kinds of cancers they target and what kinds of drugs they really want to invest in. All comes down to money.
How much money they can make. Yeah. Sure.
Ken, do you want to ask your second question? Because I don't want to butcher it.
Yeah, there is. The
Thank
question
you.
was just, thank you. The question was just about recidivir. I wondered if you could compare and contrast the strategy there to what you're talking about. Thank you very much.
So that drug, and people are probably hearing about it because Fauci talked, there was I don't know what it was, a 20, 30%, some benefit in time of recovery. So that's a different kind of drug. That's an analog of ADP. And so what it does is so how do I explain this? So when the virus infects the cell, one of the many things it wants to do is it's an RNA virus. So it wants to use its RNA to make new RNA that a new virus will want. And so that is done through an enzyme called RNA polymerase. And what this drug does, and people are still arguing about the mechanism, but it's basically an adenosine analog. So what the hope is that this analog would be put into the RNA instead of the real adenosine, and so you would screw things up. But what they think now happens is that this adenosine analog causes that polymerase to pause and to really stop working. So you can imagine if you could stop reproducing the RNA, you'd have a benefit in terms of viral infection. The thing I really wonder about is specificity and side effects. I mean, it really looks like something that would inhibit a lot of things. So I think there's going to have to be a lot of work done. And in fact, the trials that have been done so far, some patients have had pretty severe side effects.
All right, great. Well, thank you. That was super interesting and less jargon than I'm used to from crystallographers. So thanks a lot.
Ernie, do you want to go ahead with your question?
Yeah, I'm actually typing an answer to Randy Miller's question. But yeah, so Rick, you talked about iterating. And so for someone who's spending his life in his basement doing planning, during the 2 or 3 weeks of beam time, do you think you will actually have information on day 1 that will send you back to the lab on day 2 and then back with new proteins on day 3? I mean, give us some idea of the time frame.
Yeah, and you're asking a similar question to what the committee asked. And, you will have bullshit scientists out there that'll tell you, yeah, in 2 weeks, we're going to have successful drugs. This is really challenging, and I'm not trying to use that as an excuse, but getting a pseudovirus made that really works like SARS-CoV-2, but isn't infectious, that we can test, is what we're working hard on now. Mm-hmm. And to be honest, I don't know if it's going to be in a week. I don't know if it's going to be in 2 weeks. We have 3 people in molecular medicine who are really good working on this, but it's hard to know how fast that'll be. The other side of the problem is Gary Whitaker, because he's trying to make sure he can reproducibly keep SARS-CoV-2 growing in the host cells that he's using. And you would think, well, that's trivial. Everybody gets sick when they get this virus. But to keep it in a laboratory going, it's much easier to have it in an organism. Right now, we don't have a mouse model. So he's doing it, taking the real virus and infecting host cells, which he can then get a lot of information from if he can keep those cells and that virus viable. Meanwhile, we want to develop a mouse model, but that too is not going to be trivial. And just to finish it off, part of the challenge is we really need an all hands on deck approach. So because of the issue with safety, Cornell won't let me bring back 10 people. They'll let me bring back 3. And, you know, I would be ready right now to put my whole lab, which is relatively large, on this, because that's what it's going to take. And I can tell you, who's ever working on it in drug companies and hospitals have more than 2 people. So all that talk just to say, I honestly can't project, and we're pushing as hard as we can. But we'll have structures ready to go. I mean, so independent of whatever the hell we're seeing, you know, I mean, we'll have complexes that we want to look at, and whatever, if we can make the serial room temperature work, that'll be valuable for us. That'll give us a lot of useful information. We can be doing drug design while we're trying to establish how well they inhibit the virus. So.
Okay. Thank you. Excellent talk. Yeah.
Saul, would you like to go ahead with your question?
Can you hear me?
We
Yes.
can.
Oh, Christ. Hello, Saul.
Hi, Rick. Long time no see.
Yeah.
As you know, one of the themes that's been followed at Beck Chess for many years, my students and others, has been to try to understand the effect of temperature and pressure on small-scale structural changes that you have in proteins. And it was challenging because the changes that you typically see between a cryofrozen and non-cryofrozen protein is at the level of 0.2 to 0.8 of an angstrom, really small, typically not easily seen in a 3-dimensional structure. But the simple tool of looking at the RMS squared deviation of the alpha carbon backbone versus residue number picks those differences out quite nicely. You've seen those graphs. My question is, if you take the room temperature structure of your protein with and without the drug, what does that graph look like?
Well, what we have right now is a room temperature structure of the enzyme without the drug. I mean, this is a method that's still being worked out. You know, right. I mean, Aaron probably can speak more to this, but I think the initial work that was being done on cereal, aside from whatever was being done with Dwayne Miller, but on cereal was on this fluorinase, which is a much smaller protein. So the protein we're working on is on the order of, it's going to be 4 subunits, each about 60,000 or so.
Okay.
So that's a lot bigger. And I think that probably the data analysis probably becomes more complicated because of that. So we're not that far along to be able to make the comparisons yet.
Okay, well, let me just strongly encourage you when you do get the data to actually try it. I mean, both the RMS squared deviation of the alpha carbon backbone and the B factor map versus residue number turn out to be really powerful tools to see these very small scale differences.
Yeah, well, we'd like to be able to see those for sure. So that's an idea. And then what we'd like to be able to do is be able to translate the differences we see into saying, okay, how do we change the chemistry so that we can make this more potent?
Sure.
That's the ultimate. That's the ultimate.
Right. Thank you.
Sure.
Rick, I have a question from Carl here in the chat. He says, can you say how a successful treatment avoids the pathogen from evolving and surviving? Can you compare with what we have learned from treating AIDS?
Yes, so that's a good question. So, you know, there's a big effort to making it, and Saul, in full disclosure, Saul has been influencing me for the last 24 hours on the benefits of pressure in vaccine development. Mm-hmm. But so that's a big effort to make a vaccine. And you know from flu that we have to make a different vaccine virtually every year. We have to figure out which flu it is and how has it been mutated versus the year before. And Saul was pointing out last night, and there's truth to this, that was a big hang up with HIV. It was changing and it was really tough to get a vaccine. So far, and it's, you know, what I'm going to say is based on, we don't know a lot about this virus yet, but so far it looks to be not particularly mutable. That is, it's not mutating at the rates that they would have modeled for a lot of these other viruses. And so that would be good news for a vaccine. Now, the problem is that, and again, very limited data, so you got to take this with a grain of salt. no one knows yet how good are the antibodies. So when you get infected and you get over the virus, you're going to have antibodies in your plasma. And there's some that argue those antibodies don't bind very tightly. And so making a vaccine, we don't know how easy that's going to be. That's why everyone cautiously says, I think, a year to 18 months. But one of the great things about this virus versus cancer is that so there's going to be I think it's, in the end, it's going to be small molecules, hopefully a vaccine and combinations of things, just like we try to do in cancer. But the really good thing about this virus is it doesn't seem to keep, all it wants to do is get into the cell, change the metabolism, and get the hell out of the cells. Not like cancer, where the oncogenes want to take over these cells so that they're growing faster and reproducing. And so that's why when you stop one pathway in cancer, boy, it finds other ways to keep that going. This virus doesn't do that. It's a one-shot deal. So if we can get the metabolism to go back to normal, I don't think we're going to have resistance problems that we have in cancer. We'll have resistance if you have mutations in the virus that then changes the way it replicates. But I think there's some hope that it's not mutating that much. And I don't think we're going to see the same kind of resistance.
And he had a follow-up that said, please say what Whitaker's host is.
Oh, so it's probably Vera2. They use epithelial cells. And what we're using is, we're using a bunch of different hosts in our experiments. Vera2, lung epithelial cells. Those are the types that most people use for virus infection.
Okay. Thank you. And then Richard Gilliland had a question. Richard, you can go ahead.
Hi, Rick. Really fascinating talk. You in your early diagram, you showed how the cell's metabolism is changed, and the diagram sort of implied that that change took place in the mitochondria. And I'm just wondering, is that true, or is this a metabolic change that's sort of also present in the cytosol? Where's the metabolism?
Yes.
The change is going on?
There's a lot of changes to the metabolism. So what happens is that when you have a cancer-causing genes working together, they send signals that change some of the enzymes in that first pathway, the glycolytic pathway. And then they express another enzyme that is responsible for uncoupling the glycolytic pathway from what's happening in the mitochondria. So the changes that happen in the glycolytic pathway, the cancer cells use for some ATP production. You don't get as much ATP from that pathway as you do from the TCA cycle, but because of the changes in the cancer cell, there's a lot of flux. So you're making ATP to keep the cancer cells satisfying their bioenergetics, and there's some other building blocks that come from that. But they desperately need that TCA cycle. They desperately need glutamine metabolism. And the reason that we zeroed in on the glutamine metabolism, or one, it was sort of serendipitous. The compounds we found, when we finally figured out what the target was, it was that enzyme. But the other reason is that cancer cells become very much dependent on this, and normal cells, not nearly as much. There are some isolated examples, but the way cancer cells become dependent and the way that enzyme becomes active in cancer cells is how these compounds we're using block those specific activation events. So there's more than one metabolic change, but we've zeroed in on the one we think is the most specific to cancer.
I'm just curious because, as you know, there's this endosymbiosis hypothesis that mitochondria came from some ancient bacterium. And I'm just wondering whether there's enough similarity in that there might be something interesting to learn in terms of, or whether it's much more a case that the two have co-evolved and it's hard to.
I don't know the answer to that. One thing I would say that's sort of along those lines, but not to that question, and that is, I talked about these little vesicles that the cancer cells are making, that the virus is making. Turns out they show up throughout biology. So neurodegeneration, vesicles being made by cells that are undergoing neurodegeneration are very different and are contributing to that. Aging. So it turns out that that organisms, as they age, We have a mouse model for aging. And if you look at the vesicles that are being produced by some of the nervous system cells, as the animals age, they're dramatically different. So for whatever reason, these things pop up over and over. The metabolic changes are being used over and over. These vesicles are being used over and over. In terms of how mitochondria evolve, then there's this whole argument about the RNA world in terms of how life began. But I know less about that.
Yeah, I hadn't heard about the little vesicles. It's just RNA inside of them. That's really
no. There's proteins. There's microRNAs, and they get loaded very specifically. It's not like everything gets into them. So that's a pretty active area in our lab. We're trying to understand what determines what proteins get in, what RNA, and then how do they get to target sites? How do they release the cargo? And then how, in the case of, so it turns out embryonic stem cells make these vesicles, and it's for a good purpose. They shoot them out, and then you have a niche of embryonic stem cells that receive the vesicles from other embryonic stem cells, and that keeps them pluripotent. And then at the right time in development, you get vesicles coming from differentiated cells to override the embryonic stem cell vesicles, and now start the stem cells down the path of a progenitor and then ultimately a differentiated cell. So this plays out over and over. Yeah.
very cool. Thanks.
Yeah.
All right, that looks like the end of our questions. So I'm going to hand it back over to Joel.
Thanks. I just want to thank you, Rick, for a really, really interesting talk. I apologize for being the Philistine who knows no biology. I learned a tremendous amount today. Okay? And I'd like to thank everybody for coming today. But in light of the time, I'll go ahead and close the meeting. And thank you, everybody, and stay safe.